I'll take them with me to like Florida. Some other teachers.
All right, you guys.
Okay, I was watching lectures today, and we were talking. Do you guys remember when she was talking about genes, like just like genes, and then we started talking about literal genes?
Yeah.
Like it was like I don't remember what it was about, but we started talking about actual genes, and I was just listening to that conversation during the lecture.
We're like right next to it.
Okay.
Oh, it's probably listening to us. It's listening to us.
And looking at chloroplast, which is the site of photosynthesis. Remember we did the overview yesterday. The first step was the light reactions. The second step of photosynthesis is the Calvin cycle. So, and chloroplasts are where they occur. Those two steps occur. And so their thylakoids take light energy. So this is your chloroplast. We have the thylakoids, which are stacks. The stacks are called gram. And that is where the first step, called the light reactions, occur. And then the second step occurs in the liquid part of the chloroplast, which is called the stroma. So this is the liquid here, and that's where the second step, which is your Calvin cycle, occurs. And so we did the overview of that. The light reactions need to occur before the Calvin cycle. Can somebody tell me why the light reactions need to occur before the Calvin cycle? Roman? That's right. So they break down water. Remember, light comes in and is absorbed by the pigments, which are found in the membranes of the thylakoid. And then water comes in, and water is split. And the oxygen part of water leaves as O2 in the atmosphere, but the hydrogen part of water meet up with NADP+, and then they come out as NADPH. So you have this electron carrier carrying electrons that were once part of the hydrogen of the water molecule. And the second thing that happens with the use of the sunlight is that it provides energy to put the phosphate onto ADP to make ATP. And so therefore, the light reactions need to happen first because these two things are used in the Calvin cycle. And the Calvin cycle is where we get our sugar, our glucose. And so we need those two things to make glucose, also with CO2. And so the light reactions occur first. So this is the purpose of the light reaction. So the thylakoids form light energy absorbed by the pigments in the thylakoid, and make ATP and NADPH. So that's what they're talking about here. That's the light reactions. So let's look at light and talk about the properties of light. So the nature of sunlight. Light is a form of what we call electromagnetic energy. This is also called electromagnetic radiation. And like other electromagnetic energy, light travels in rhythmic waves. And so we describe different parts of the electromagnetic spectrum according to the wavelength. The wavelength is the distance between crests of a wave. So when we look at a wave, it's like this. This is how light travels in waves. And this is a crest, and this is a crest. And the distance between those crests is what we call a wavelength. So you have higher wavelengths, the farther apart those crests are spread out. Shorter wavelengths, the closer that they are. And then the wavelength determines the type of electromagnetic energy. So, another thing that you need to understand is that light also behaves as though it consists of particles called photons. So later on in this chapter, we'll talk about photons of light. So you just need to understand that a photon is like a package of light energy. When you see that terminology. So you have a picture here that shows you, the next one shows you a picture of the electromagnetic spectrum. So the electromagnetic spectrum is the terminology used to describe the entire range of all electromagnetic energy or radiation. Whoops. And visible light is just a small portion of that electromagnetic spectrum. Visible light consists of colors, we can see, including wavelengths that drive photosynthesis. Hence the name visible light, we can see it. Okay? So this picture here is showing you the electromagnetic spectrum. And so what we have here, these are the different types of radiation here. And on the top, notice there it says 10 to the negative 5 nanometers and 10 to the third meters. And so we have radio waves. So the numbers at the top there are talking about the wavelengths, the distance between the crests. And so radio waves have a lot of distance between the crests. So they travel, so you know the distance between them are really far. All right. Then we go to 10 to the negative 5 nanometers for gamma waves, and they're like this. All right. So the distance between them are really, really short. And so that's kind of the trend here. We go from longest wavelength, greater distance, to very close here. And so visible light is just a small section kind of in the center of this electromagnetic spectrum. And this is the light we can see. This is white light. White light is comprised of a bunch of different wavelengths of light that collectively make white light. So white light is all these different wavelengths or colors of light. Each wavelength of light appears to us as a different color. But when all of these wavelengths together, all these different colors together, we see white light, okay? And so that's our visible light, and this is what's used for photosynthesis, is the visible light, the light that we can see. So this is the white light shining from the sun. Okay, so then looking at here, then we have shorter wavelength to longer wavelengths, so that's kind of the same trend here. But I want to show you the correlation. Shorter wavelengths have higher energy, longer wavelengths, lower energy. All right, so there's this. So when we're looking at this, yours is not color coded. So you might want to. It's right here, violet, and then goes to blue, then to green. So you have some, frame of reference, because I doubt that anybody has these wavelengths of light of various colors memorized. And so you kind of get an idea of what wavelength of light. And so it's kind of the Roy G. Biv for the rainbow. Going backwards. You know Roy G. Biv?
Yeah.
All right. So, because red has the highest energy. So this is our electromagnetic spectrum, and so we're going to be talking about visible light here. So white light shines on the plants and go into the cells, go into the chloroplast, and it's these pigments that absorb it. So let's look at pigments here. So what is a pigment? They're substances that absorb visible light. Different pigments absorb different wavelengths. And pigments, we'll see in just a minute, there's a molecule, they're a chemical they're made out of carbons, hydrogens, oxygens, and so on there. A physical molecule has a specific chemical makeup. And so different pigments have different structures and therefore absorb different wavelengths or colors of light, the visible spectrum. And so wavelengths that are not absorbed are reflected or transmitted. And so leaves appear green because chlorophyll reflects and transmits green light. So why things appear have a specific color is because what's being reflected back to our eyes. And so if somebody has a blue shirt on, it appears blue because white light from our light bulbs is shining on the shirt. White light is the whole Roy G. Biv spectrum. Certain wavelengths of light of that Roy G. Or different colors of light are absorbed into the shirt, into the pigment of the shirt, and some is reflected back. And so blue, if something appears blue, it's because blue light is being reflected back to us and it appears blue. So it's not absorbing blue light. So the dyes in our clothing are pigments, and so they reflect and absorb different wavelengths of light. So when something is black and when it appears black, does anybody know what that means? Joe? Everything's absorbed. So all of the wavelengths of the white light, all those different colors are absorbed. And white It's just the opposite. All of them are reflected back, and that's when something appears white. So think about this. This is why it's warmer. Like if you're going to go work outside in the summertime, it's best to wear not a black shirt, but a white shirt, because the black shirt, the dye in there is going to absorb all the wavelengths of light. That's energy. It's going to create heat and make you hotter. All right, same reason why black cars get hotter than white cars and so on. And that's also why we can't see anything if something is if we're in a pitch dark room where there's no light coming in. That means light isn't reflecting off of anything, so we don't see any reflection whatsoever, and it appears dark. All right, so you have to have some light coming in to reflect off of surfaces to be able to see anything in the first place. All right, so what would happen to a plant if we tried to grow the plant under a green light? What would happen to the plant if we tried to grow the plant under a green light? That's right. So it's not, yeah, so it reflects green light. So if you're only giving it the wavelength of green light, as Max said, it's going to reflect back, so it's not going to absorb that green light. Is it going to be able to do photosynthesis?
No.
No. If it can't do photosynthesis, what is it not making?
Glucose.
Glucose. If it doesn't have glucose, what can't happen?
Cellular respiration.
Cellular respiration. If we don't have cellular respiration, we're going to run out of
ATP.
ATP for energy. And then so what's going to happen to the plant over time? It's going to die. Absolutely. So don't grow your plants in a freezer. All right. So, moral of the story. So this is showing you the chloroplast and showing you visually the white light is the whole spectrum or color of light, all the different colors of light. It hits the thylakoid, and why they're showing you the thylakoid is because that's where the pigments are in the membrane of the thylakoid. So if chlorophyll is the pigment, it's going to absorb certain wavelengths of light, and then green light is going to be reflected and transmitted. So this is green light, and so you might want to write there that this is green and this is green, because mine is color coded, yours is not. So it's reflecting and transmitting green light. What kind of light is it absorbing then? So right now we know that green light is reflected, and we know that then we can pretty much assume that green light isn't used for photosynthesis. But what color or wavelength is used for photosynthesis? So then we're going to look at that. So in order to study pigments, we use a machine called a spectrophotometer. This measures a pigment's ability to absorb various wavelengths of light. The machine sends light through pigments and measures the fraction of light transmitted at each wavelength. And so this is what we have here going on. So this happens inside of a machine. So the machine, a spectrophotometer, is just like a box that sits here at the table. It's about yay big like this, right? It's not very big. And on the top of it, you have a little opening that you can put a test tube in. And so what you can do is put pigments, a solution of a certain pigment in a test tube, and put it into this machine. That's what they did here with chlorophyll. So we put the test tube in this machine. Now inside the machine, we have a source of white light. So here's the white light bulb as our source of white light. We turn it on, the white light comes on, the white light shines and hits this prism. The prism separates the white light into all the different wavelengths or colors of light, which is what we see here. Then on the outside of it, there's a dial that allows you to pick what wavelength out of all the visible light wavelengths from red to violet, what wavelength of light do you want to beam at your sample? All right, so you turn this dial, and so notice that there's a filter here, and this hole moves up and down so we can beam red light on it, orange light, and so on and so forth. So they chose green light. So here's green light being beamed at your sample. So now what we want to see is green light absorbed or reflected or transmitted. So when this green light hits the chlorophyll, chlorophyll doesn't absorb green light. So what happens is a lot of that gets transmitted and goes right through the sample because it's not absorbed. And so then there's this photoelectric tube that measures how much light is actually hitting, how much light is being transmitted. and it communicates that to this dial here, and this is measured in transmittance, so it's almost 100%. So out of the light that's being beamed at our chlorophyll, almost 100% of it is transmitted. So if almost 100% is transmitted, then that means there's very low absorbance, because they're opposite. So if it's being transmitted, it's not being absorbed. If it's being absorbed, it's not being transmitted. Does that make sense? All right? Yes.
So since green, like if you look at a color wheel, green is comprised of blue and yellow. So
Light is different than pigments. Like, all right, so it's a different thing. So then we look at this, and so we get a high transmittance, low absorbance, which is what we would expect with chlorophyll. So what does this prove? It just proves that green light is not absorbed by chlorophyll. So then what we want to do is see what is absorbed by chlorophyll. What color of light would be absorbed by chlorophyll, because that color of light most likely is involved in photosynthesis. So then so we can change our dial and change the filter and beam any color of light we want to at it. We can do it for all of them. And so the next picture shows you doing it with blue. So we choose blue, it beams blue light at the sample. Very little blue light is transmitted. So we have a low transmittance here, which then correlates to a high absorbance. That means why is there not very much going through? Because the chlorophyll absorbed a lot of that blue light, all right, and didn't allow it to go through. So, have we proven though that blue light is needed is used in photosynthesis? We've proven that? No, what we've proven is that chlorophyll absorbs blue light. We can make a hypothesis and a best guess that blue light then is probably used for photosynthesis, but from this experience, like doing this, we don't know that. And so, I'm going to show you how we know that for sure. All right, so this process here, using this spectrophotometer, allows us to make what we call an absorption spectrum. It's a graph plotting the pigment's light absorption versus wavelength. The absorption spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis. So that's what we just talked about here with the blue. A lot of it gets absorbed. And red light work best for photosynthesis. If they made the conclusion that red light works best for photosynthesis, if you beamed red light at this, what would your results be? Yeah, close to zero as well. That means that a lot of the red light would be absorbed and therefore used for photosynthesis. So then write the next one, the action spectrum. An action spectrum profiles says the relative effectiveness of different wavelengths of radiation in a driving process. So here we know that the chlorophyll absorbs violet, blue, and red light, and we can hypothesize then that those are the wavelengths that work best for photosynthesis. This shows you those wavelengths and how it actually affects those different wavelengths affect a driving process. The process that we're looking at is photosynthesis. So how do these wavelengths affect photosynthesis if they're being absorbed? So here's your absorption spectrum. This is what the graph looks like after doing all of that work with spectrophotometers. So usually the yellow line doesn't copy very well, so you might want to draw that in. Do you see that? The yellow doesn't. Sometimes it appears like almost like a white. Do you see it or no?
No.
No, not at all. All right, go ahead and draw that in, and then it stops here and it's zero for the rest of the. And this is also figure 10.9a in our textbook. Pretty much said that for the video, so people who can't watching the video can't see the yellow line. Okay, looking at here? Here are various wavelengths of light, and we're graphing that versus the absorption of light, and we're looking at different pigments. So we have 3 different pigments. Notice it says chlorophyll A and chlorophyll B. So there are actually 2 different types of chlorophyll, and they have a slightly different chemical structure, which makes them absorb slightly different wavelengths of light. Maybe put in parentheses behind chlorophyll A, put that this is the main photosynthetic pigment. It's the main photosynthetic pigment. So if we just look at chlorophyll A, this main photosynthetic pigment, what they did is they took a test tube of chlorophyll A and put it in the spectrophotometer and beamed every single wavelength of light at it and recorded how much is absorbed. And so what they found is that, notice here, we get a peak here. So that's in that violet-blue range. Hence, back over here, when we wrote that the absorption spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis, that's where we're getting it at, because in chlorophyll A, we get a peak of absorbance, a high absorbance in that violet-blue range, and then notice it goes down and it stays close to 0 all through the green-yellow range, and we get another peak here towards the red. So these peaks mean that there are high absorbance, which means that most likely those are the light colors or the wavelengths of light that are useful for photosynthesis. These 2 pigments right here are called accessory pigments. They absorb slightly different wavelengths of light than chlorophyll A. So one of the reasons for that is it allows the plant to absorb a greater variety of wavelengths. And so we'll look at more specifically in just a few minutes on what they do. So if you look at chlorophyll B, which is this lighter green line, we have a peak that's slightly different, slightly offset than the chlorophyll A. So it absorbs slightly different wavelength of light. It's low through the green-yellow range, and we get another peak here that's just slightly different than chlorophyll A, and gives it a wider variety of wavelengths of light that it actually absorbs. And then lastly, the carotenoids. The carotenoids are a group of pigments, and you can see that they have a couple of peaks here, and then also drop down here, and they also absorb slightly different wavelengths of light. And so this is your absorption spectrum. And so from this, we can hypothesize the wavelengths of light that are useful in photosynthesis, but we haven't proven it. So in comes the action spectrum. The action spectrum, the x-axis is the same as the last picture. So it's supposed to be, yours is kind of on top of one another, and it doesn't, they assume that you know that, and so they should assume that here. So we should write it here, wavelength. So we go from violet to red, just like the last picture. So it's the same x-axis. The y-axis, though, changes. The y-axis is this action. What's the action? We're looking at photosynthesis. So what's the effects of the different wavelengths of light on photosynthesis? So that's what we have here on the y-axis, the rate of photosynthesis measured by oxygen release, because O2 is released when plants do photosynthesis. And we see a higher rate of photosynthesis in the violet blue and a higher rate of photosynthesis in the red and a lower rate in that green-yellow area. So how do they know this? And so in comes a gentleman named Thomas Engelmann. This is a famous experiment that he did here with bacteria and algae. In his experiment, he exposed several different segments of filamentous alga. That is algae that grows in fresh water, to different wavelengths of light. And filamentous means it's like thread-like. All right, so it's a long piece of algae. And what he did is, as he beamed different wavelengths of light at this, areas receiving wavelengths favorable to photosynthesis produced excess O2. Because if they get the right light, they're going to do photosynthesis, they're going to absorb it and do photosynthesis, and one of the products of photosynthesis is producing oxygen and releasing oxygen. Then he used, the second organism he used is aerobic bacteria, clustered along the alga as a measure of oxygen production. Aerobic means, what do you think aerobic bacteria means?
It has access to oxygen.
The bacteria that need oxygen to do cellular respiration. So they can't survive in anaerobic conditions. All right, so the more oxygen, the better. All right, so this is what he did. All right, and so this is the picture here, but let me explain, to understand this picture, you kind of have to understand a little bit about bacteria and growth in the algae. So I learned in the other hours that very few people have ever grown bacteria. Is that true in here? You guys ever grown bacteria?
No.
All right.
I remember in like third grade when we took bread and like rubbed it on the gym floor and then we rolled.
You roll, but that's different than bacteria.
That's disgusting.
So,
That's like
So,
horrifying.
that's not as bad as when the ninth grade used to be here at the high school, and I taught just ninth grade biology, and we did an experiment where I had them go and swab different areas around the school for bacteria. You can just use Q-tips and go to different spots. And you grow bacteria. Let me explain how you grow bacteria. You use Petri dishes. You guys know what Petri dishes are? All right, Petri dishes, you put in something on the bottom called agar. Agar is kind of, it's denser than Jell-O, but it's kind of gel-like, all right, and it contains food for the bacteria. So you have food in this agar, and what you can do is you can't see bacteria. So if you want to test if bacteria is growing on something, you can take something like a Q-tip and rub it on the surface, and then go to your Petri dish and rub gently the Q-tip on top of the agar. And so any bacteria that's on there will get transferred to the agar. You can't see the bacteria, but it'll get transferred. And I throw the Q-tip away, put the top on. And I had my students seal. I had them tape the tops on it, because once we started growing bacteria, we wouldn't get anybody sick. So, and then we never opened it back up. And then you can't see anything growing on there, and you put it into an incubator, and the bacteria are very, very happy in the incubator. They're warm and they have food. And so if you have one little bacteria sitting there, you can't see it. So there's that little bacteria. You can't see it when you first put it on, but in this nice environment, that one bacteria divides, and then you have two bacteria. Then they divide, and then you have four bacteria. And within 24 hours, you literally have millions of bacteria. And you can see when millions of bacteria are clustered together, you can see them. You see this little kind of circle. And when you look down on it, it literally looks like a little circle. And that is a colony of bacteria. It's millions of bacteria growing on there. And so you can see it growing and so on. So that's how you grow bacteria. And so and it was interesting, just a little tidbit. I let students pick what areas, things that they wanted to grow. Somebody always picked the toilet. All right, always, never failed. But it was interesting that kids also chose the spigot, the spout where the water comes out of the drinking fountain.
Oh no.
Drinking fountain won every time versus the toilet in terms of bacteria.
Awesome.
It's really gross. And close with that is people who do it with their hands. Right? Yeah, or the tabletops.
Oh.
Just to give you a little something.
I know, like,
So we need to pull out the lights after this discussion. All right, so when we're looking back to this experiment, what he did, if I were to look down at the top of the Petri dish, so we're looking at a circle here and looking at the surface of the agar, what he did is he took this, he took the algae, which looks like a thread, and he put it on the surface of the agar, and then he took bacteria and swabbed, aerobic bacteria that need oxygen, he swabbed the surface of the agar. And then what he did is, so you can't see any of the bacteria right now because they're so small, and then what he did is started beaming different wavelengths of light on the algae. So that's what this picture is showing you. So he beamed right here, purple light. This part of the algae got blue light. This part of the algae got green, and so on and so forth. And so the areas that the algae was doing the most photosynthesis, which happened to be in the blue and the purple range, started producing oxygen. And in the red range, started producing oxygen because they're doing photosynthesis. In the green-yellow range, they weren't really absorbing those wavelengths of light. So the algae in this range here wasn't really producing oxygen because it's not really doing photosynthesis. It wasn't getting the right wavelength of light. So guess what? The bacteria that didn't get a lot of oxygen didn't divide very much, and it didn't see very much growth on the agar. Here and here, he did. So he saw, literally growing on his petri dish, lots of colonies of bacteria congregating in the areas where photosynthesis was going on. If you notice the shape of the algae growth here, looks just like, well, almost just like the action spectrum. So we have the high growth here and here. And so that was the experiment that proved that they always hypothesized that blue, violet, and red light was helpful in photosynthesis, and that proved. Okay? Alrighty. So let's look more specifically at chlorophyll A, all right, and the pigments. So now we know that what color of light, or what wavelengths of light it absorbs. And so chlorophyll A, we talked about being the main photosynthetic pigment, and then accessory pigments such as chlorophyll B. What that does is it broadens the spectrum used for photosynthesis. Why? Because chlorophyll B uses a slightly different wavelength, or absorbs, I should say, a slightly different wavelength than chlorophyll A. The second accessory pigment, called carotenoids, which is a class of molecules, a bunch of different ones, their job is to absorb excessive light that would damage chlorophyll. So they absorb some light energy so that chlorophyll, the plant doesn't absorb too much energy and damage the main molecule for photosynthesis. So two kind of reasons for accessory pigments. And that's just, I just showed you, I've just included this in there to show you that chlorophyll is truly a molecule with a specific shape and structure, and it's made out of atoms. It has a hydrocarbon tail here, and so on. And also to show you up, it kind of is cut off in my picture here, but up in the upper right-hand corner, it highlights the one chemical group. Yours is kind of highlighted in a lighter color. The one chemical group that's changed between chlorophyll A and chlorophyll B. Everything else in the molecule, all of this part is the same between the two, and just one little chemical group has changed. And that one little chemical group changes the structure slightly so that it absorbs different wavelengths of light. So that also goes with the theme of structure affects the function, all right, in terms of absorption. Okay, so what happens when chlorophyll absorbs light? So, when a pigment absorbs light, it goes from what we call a ground state to an excited state. And the excited state is unstable, so it doesn't stay at the excited state very well, or very long. What does it mean to be excited? We're actually talking about electrons in the atoms of the molecule. So when excited electrons fall back to the ground state, photons, which are light, so this is light, are given off, which we call, the afterglow that is called fluorescence. And so if illuminated, are given light, and an isolated solution of chlorophyll will fluoresce, giving off heat and light. So what does this mean? So if we take the chlorophyll out of a plant, so if we isolate and take chlorophyll out of the plant so it's not in the plant anymore, and put it in a beaker or something like that, so we have a solution of chlorophyll, and we beam light at it, what's going to happen? So here's your chlorophyll molecule. It's not in the plant anymore. All right, so your light, here's your photon of light. So this is light energy. The chlorophyll molecule absorbs it. What happens is the chlorophyll molecule, I just showed you the structure, has carbons, hydrogens, oxygens, and so on. It has all these atoms that are made out of electrons that are in various energy levels. And so what happens is when the chlorophyll molecule absorbs the light energy, the electrons in the atoms become excited. Do you guys know what that means? Anybody have an idea? When the electrons get excited?
They like jump from a warp level. Right?
They do what?
They like jump from a lower level into a higher level.
That's right. All right, so when we're looking at, let me draw here, so a generic atom has protons and neutrons in the nucleus, and it has the electrons spinning around, so there's two electrons, and so on. So when an electron becomes excited, it jumps, this is the next, oh no, well, it goes to the next orbital there, the next energy level there. All right, so that's what it means to become excited, but it doesn't stay there long. So what happens is it'll drop back down. So it absorbs the light energy, the electrons become excited in the atoms, and then the excited electrons drop back down to the ground state. Now, in order for it to become excited, though, it had to absorb energy. So when the energy is absorbed and it becomes excited, to go back to its original position, that energy has to go somewhere. So that energy that it absorbed to make it excited to begin with is now released into the atmosphere. And then what happens is it's released in the form of heat energy and light energy, and that's the fluorescence. And so what actually happens, this picture is a picture of a flask of isolated chlorophyll that has been, they shined white light on it, and all the chlorophyll molecules, this is what's going on. So the electrons are becoming exciting and it drops back down. And so the fluorescence of chlorophyll is actually red. It fluoresces red, and if you feel the flask is actually warm. All right, and so that's what happens with chlorophyll. But this is an isolated chlorophyll molecule. Now, if we put the chlorophyll back into and talk about chlorophyll in a plant, chlorophyll in a plant does the same thing. It absorbs light energy. The electrons get excited, but do the plants fluoresce? No, our plant, that would be kind of cool, but our plants do not fluoresce red. We don't have glowing red plants. And so what happens is the electron dropping back down, that doesn't happen, because that's the part that makes it fluoresce. So the chlorophyll molecules in plants, the electrons get excited, but they never drop back down, and therefore we don't have fluorescence. And so what happens is we have a molecule, or the plants have a molecule called the primary electron acceptor, that once the electrons get excited, they accept the electrons and take them as part of themselves, and therefore the electrons never drop, and we don't have glowing plants. All right, question?
Is that like how glow in the dark stuff works? Is it?
It depends on what kind of, like,
So like if you just have like,
not a glow stick.
no, I'm talking about like if you have, you know, it's like the translucent
Yes.
things that
Yes.
you shine.
Yes. Alrighty, is that what you're gonna ask? Okay. Alright, so here we'll stop, alright, because I think we get out at 58, so I'll let you go here at 55.
That'd be good.